Ketamine has been long recognized for its anesthetic effects and has gained recent popularity as a method of treatment for major depressive disorder in individuals who are resistant to typical SSRI pharmacotherapy. Clinical ketamine administrative treatment results in more rapid and sustained symptom effects than those seen in SSRI treatment. Current understanding of how ketamine produces these antidepressant effects is unknown, but it is believed to rely on inhibition of NMDA receptors, resulting in disinhibition of glutamatergic neurons, leading to a subsequent rise in the release of glutamate. Human treatment trials for major depressive disorder show that other antagonists to NMDA receptors are not as effective as ketamine in reducing symptoms of major depressive disorder. In the present study, we examine alternative mechanisms in the antidepressant action of ketamine and its metabolites. To do this, mouse models are randomly assigned to a variety of tests that measure antidepressant-like responses to different drugs. Some of the major tests have been included here. The first step of our study involved identifying the possibility of an NMDA receptor independent route of antidepressant action of ketamine. If the antidepressant actions of ketamine are to rely on inhibition of NMDA receptor action, then these must be able to be repeated using MK801, another NMDA receptor antagonist. Mice were given saline, ketamine, or MK801 and were then subjected to the forced swim test, in which mice are to swim in room temperature water for 6 minutes. Immobility time was measured by time spent inactively floating, which is interpreted as a lost hope to survive. We found that while both drugs had a significant antidepressant response when tested 1 hour after drug administration, antidepressant effects were only sustained in ketamine-treated mice. In a social stress test where experimental mice are conditioned to a novel mouse pre-screened for aggressive behaviors, mice that were determined to be depression susceptible exhibited an increase in social behavior when presented to another stranger mouse after treatment with ketamine, but not MK801. These two findings together implicate that inhibition of NMDA receptor action is not sufficient for antidepressant actions of ketamine treatment. Analysis of the forced swim test data was indicative of a greater sustained antidepressant effect of ketamine in females when compared to males. This led us to examine the pharmacokinetic profile of ketamine and its metabolites between sexes. In doing this, we found that female brain tissues had threefold higher concentrations of the metabolite hydroxynorketamine, or HNK, compared to that of males. This relationship led us to dig deeper into the pharmacokinetic and dynamic profile of ketamine metabolites. To start, ketamine was deuterated into 6,6-deuteroketamine as to reduce rate of ketamine metabolism. As seen in 3A and B, administration of deuterated ketamine resulted in greater antidepressant effects in the forced swim test as well as the learned helplessness test, outlined in the methods section, confirming that metabolism of ketamine into its metabolites is sufficient for the antidepressant effects of ketamine. Further analysis into the effects of the individual ketamine metabolite enantiomers 2S6S-HNK and 2R6R-HNK, whose structural difference may be seen in 3E. Administration of both ketamine and 2R6R-HNK resulted in significantly greater antidepressant effects than 2S6S-HNK in both forced swim tests and the learned helplessness test, implicating the R enantiomer as the active metabolite for antidepressant action. We performed some in vitro tests on mice brain slices and found that application of 2R6R-HNK induces an increase in AMPA-mediated EPSPs, which was sustained through drug washout. And important to note, AMPA is just another glutamate receptor. To test this in vivo, we treated mice with the AMPA antagonist MBQX 10 minutes prior to ketamine or 2R6R-HNK administration. Mice were then subjected to the forced swim test, where it was shown that pretreatment with MBQX resulted in decreases to both acute and sustained antidepressant actions of both ketamine and its metabolite. Additionally, we were able to determine that treatment with 2R6R-HNK does not induce addictive potential like treatment with ketamine, as demonstrated by our drug discrimination and self-administration tests. In summary, we identify a novel route for the antidepressant actions of ketamine, identifying the innocuous metabolite 2R6R-HNK as the active molecule in facilitating antidepressant behavior. These findings hold promising potential for next-generation fast-acting antidepressant treatments. Thanks for watching.